Everyone, welcome to the video lecture from Unit 2, Module 3.2.3. In this one, we're going to talk about hemostasis. As always, learning objectives are right there. Make sure you follow those for what will be on the unit exams and the lecture quizzes. So let's get started. So the process of hemostasis is the stopping of bleeding. Okay? It halts the loss of blood to the walls of damaged vessels. At the same time, it establishes a framework for tissue repairs. Hemostasis has three phases: the vascular phase, the platelet phase, and the coagulation phase. The boundaries of these phases are somewhat arbitrary, but in reality, hemostasis is a complex cascade or a chain reaction in which many things can happen at once, and all of them interact to some degree. So cutting the wall of a blood vessel triggers a contraction in the smooth muscle fibers of the vessel wall. This local contraction of the vessel is a vascular spasm, which decreases the diameter of the vessel at the site of the injury. Such a constriction can slow or even stop the loss of blood to the wall of a small vessel. The vascular spasm lasts about 30 minutes or so, and that's a period called the vascular phase of hemostasis. During the vascular phase, there's a couple changes that take place in the endothelium of the vessel at the injury site. The endothelial cells will contract and expose the underlying basement membrane to the bloodstream, and the endothelial cells will also begin releasing chemical factors and local hormones. We'll discuss several of these factors, including ADP, tissue factor, and prostacyclin in later sections when we get there. But endothelial cells also release peptide hormones called endothelins. And these local hormones, one, stimulate smooth muscle contraction and promote vascular spasms, and two, they stimulate the division of the endothelial cells, smooth muscle cells, and the fibroblasts to accelerate the repair process. Continuing on, the endothelial plasma membranes become sticky. A tear in the wall of a small artery or vein may be partially sealed off by the attachment of endothelial cells on either side of the break. In small capillaries, endothelial cells on opposite sides of the vessel may stick together and prevent blood flow along the damaged vessel. The stickiness is also important because it helps platelets to attach as the platelet phase gets underway. So the attachment of platelets to the sticky endothelial surface, to the basement membrane, and to exposed collagen fibers marks the start of the platelet phase of hemostasis. The attachment of platelets to exposed surfaces is called platelet adhesion. As more and more platelets arrive, they begin sticking to one another as well. This process, called platelet aggregation, forms a platelet plug that may close the break in the vessel wall if the damage is not severe or if the vessel is relatively small. Platelet aggregation begins within 15 seconds after an injury occurs. As they arrive at the injury site, the platelets become activated. The first sign of activation is that they become more spherical and develop cytoplasmic processes that extend towards the nearby platelets. At this time, the platelets begin releasing a wide variety of compounds. These compounds include 1. Adenosine diphosphate, or ADP, which stimulates the platelet aggregation and secretion. 2. Thromboxane, A2, and serotonin, which stimulate vascular spasms. 3. Clotting factors, or proteins that play a role in blood clotting. 4. Platelet-derived growth factors, or PDGF, which is a peptide that promotes vascular repair. And 5. Calcium ions, which are required for platelet aggregation and in several steps in the clotting process. The platelet phase proceeds quickly, as ADP, thromboxane, and calcium ions released from each arriving platelet stimulate further aggregation. This positive feedback loop ultimately produces a platelet plug that will be reinforced as clotting occurs. However, platelet aggregation must be controlled and restricted to the injury site. Several key factors limit the growth of the platelet plug. 1. Prostacyclin, which is a prostaglandin that inhibits platelet aggregation and is released by endothelial cells. 2. Inhibitory compounds released by white blood cells entering the area. 3. Circulating plasma enzymes that break down ADP near the plug. And 4. Compounds that, when abundant, inhibit plug formation, for example, serotonin, which at high concentration blocks the action of ADP. And 5, the development of a blood clot, which reinforces the platelet plug but isolates it from the general circulation. The vascular and platelet phases begin with a few seconds after the injury. The coagulation phase, or blood clotting, does not start until 30 seconds or more after the vessel has been damaged. Normal blood clotting depends on the presence of clotting factors, or procoagulants, in the plasma. Important clotting factors include calcium and 11 different proteins. Many of the proteins are proenzymes or inactive enzymes, and when converted to active enzymes, these proteins direct essential reactions in the clotting process. During the coagulation phase, enzymes and proenzymes interact successively. So the activation of one proenzyme commonly creates an enzyme that activates a second proenzyme, and so on in the cascade. The extrinsic pathway begins outside of the bloodstream in a vessel wall. The intrinsic pathway begins inside of the bloodstream with the activation of a circulating proenzyme, and these two pathways converge at the common pathway. So the extrinsic pathway begins when damaged endothelial cells or peripheral tissues release factor 3, also known as tissue factor, or TF. The greater the damage, the more tissue factor is released, and the faster the clotting takes place. Tissue factor then combines with calcium and other clotting factors, so like factor 7, to form an enzyme complex capable of activating factor 10, the first step in the common pathway. The intrinsic pathway begins when the activation of proenzymes, usually factor 12, exposed to collagen fibers at the injury site or a glass surface of the side of our collection tube or something like that, whatever it may be. This pathway proceeds with the assistance of PF3, a platelet factor released by aggregating platelets. Platelets also release a variety of other factors that accelerate the reactions of the intrinsic pathway. After a series of linked reactions, activated factors 8 and 9 combine to form an enzyme complex capable of activating factor 10. The common pathway begins when enzyme complexes from either pathway activate factor 10. Activated factor 10 activates a complex called prothrombin activator. Prothrombin activator converts the proenzyme prothrombin into the enzyme thrombin. Thrombin then completes the clotting process by converting fibrogen, a soluble plasma protein, to insoluble strands of fibrin. Interactions among the pathways. When a blood vessel is damaged, both the extrinsic and the intrinsic pathways respond. The extrinsic pathway is shorter and faster than the intrinsic pathway and is usually the first to initiate the clotting process. In essence, the extrinsic pathway produces a small amount of thrombin very quickly. This quick patch is reinforced by the intrinsic pathway, which later produces more thrombin. If you need something to kind of relate this to, think about the different steps in ATP production, where in the first stage you can produce 2, and you can produce that very quickly and rapidly when you need that ATP. And then in the latter stages, when you have more oxygen available, you can go into the Krebs cycle and cellular respiration and produce far more. It's the same thing here. So when you need it quickly, you're going to go through the extrinsic pathway. And then when you need to produce more thrombin at the site, you're going to use the intrinsic pathway. So the time required to complete the clot formation varies with the site and the nature of the injury. And in tests of the clotting system, blood held in fine glass tubes normally clots in about 8 to 18 minutes, which is the coagulation time. So a small puncture wound typically stops bleeding in about 1 to 4 minutes or so. So the activity of the common pathway stimulates both the intrinsic and the extrinsic pathways. This positive feedback loop accelerates the clotting process, and the speed can be very important in reducing blood loss after a severe injury. Thrombin generated in the common pathway stimulates blood clotting by 1, stimulating the formation of the tissue factor TF, and 2, stimulating the release of PF3 by platelets. So, how is clotting controlled or stopped? Blood clotting is restricted by substances that either deactivate or remove clotting factors and either stimulatory agents from the blood. Examples include the following. Things like normal plasma contains several anticoagulant enzymes that inhibit clotting. 1, antithrombin 3, inhibits several clotting factors, including thrombin. Heparin, which we mentioned in the last video, and is a common anticoagulant that's given in hospitals and things like that, is a compound that's released by the basophils and the mast cells. It's a cofactor that accelerates the activation of antithrombin 3, and heparin's also used clinically, like I said, to impede or prevent clotting. Thrombomodulin is released by endothelial cells, and this protein binds to thrombin and converts it into an enzyme that activates protein C. Protein C is a plasma protein that activates several clotting factors and stimulates the formation of plasmin, an enzyme that gradually breaks down fibrin strands. Prostacyclin, released during the platelet phase, inhibits platelet aggregation and opposes the stimulatory action of thrombin, ADP, and other factors. Other plasma proteins with anticoagulant properties include alpha-2 macroglobin, which inhibits thrombin, and C1 inactivator, which inhibits several clotting factors involved in the intrinsic pathway. The clotting process involves a complex chain of events, and disorders that affect any individual clotting factor can disrupt the entire process. For this reason, managing many clinical conditions involves controlling or manipulating the clotting response. Aspirin, for example, is a common drug that interferes with the blood clotting process and prolongs bleeding time. It acts by inhibiting platelets from producing prostaglandins and thromboxane A2, which stimulates new platelet activation and aggregation, and is also one of the reasons that when you go to the doctor or you've had an injury or anything like that, they ask you, are you on any anticoagulants such as aspirin or like heparin, things like that. So, calcium ions and vitamin K affect almost every aspect of the clotting process. All three pathways, the intrinsic, the extrinsic, and the common, require calcium. So, any disorder that lowers plasma calcium concentrations will impair blood clotting in turn. Adequate amounts of vitamin K must also be present for the liver to synthesize four of the clotting factors, including prothrombin. Vitamin K is a fat soluble vitamin present in green leafy vegetables, grains, and organ meats that's absorbed with dietary lipids when we obtain roughly half of our daily requirement from the diet. Bacteria in the large intestine manufacture the other half for us. So, a vitamin K deficiency may arise from a diet that's inadequate in fats or in vitamin K, or disorder that affects fat digestion and absorption, such as problem with bile production, or prolonged use of antibiotics that kill normal intestinal bacteria. So, a deficiency in vitamin K will cause the eventual breakdown of the common pathway due to a lack of clotting factors, and ultimately, the deficiency can deactivate the entire clotting system as a whole. Clot retraction, or synergis, pulls the torn edges of the vessel closer together. This process reduces residual bleeding and stabilizes the injury site. The process also reduces the size of the damaged area, making it easier for fibroblasts, smooth muscle cells, and endothelial cells to complete repairs. As the repairs proceed, the clot gradually dissolves in a process called fibrinolysis. And the process begins when two enzymes activate the proenzyme plasminogen. These enzymes are thrombin, produced by the common pathway, and tissue plasminogen activator, or TPA, released by damaged tissue of the site of the injury. The activation of plasminogen produces the enzyme plasmin, which begins digesting the fibrin strands and eroding the clot. To perform its vital functions, blood must be kept in motion. On average, a red blood cell completes 2 circuits around the cardiovascular system about each minute or so. Blood begins to circulate around the third week of the embryonic development phase and continues throughout life. If the blood supply is cut off, dependent tissues may die in a matter of minutes.